Assalamualaikum, I am Hanis Khariah and today I will present my study about the isolation of foodborne pathogens from blowflies Lucilia sericata collected from mini wet market at Shah Alam, Selangor. I am under supervision of Shamsul Bahrain Ghulam Ali. To start with, I will briefly introduce the research background of my study. Firstly, flies live closely with bacteria as they breed and feed in places that are rich in microorganisms. Secondly, flies play a crucial role in cross-contamination between filthy environment and food sources. Thirdly, its ability to fly approximately up to several kilometers in days have made it to become an agent in spreading diseases. Moving on, there are four significance of the study. Firstly, flies may carry pathogens and spread around the human populations. Secondly, this gives a demonstration of food or raw materials contaminated by flies are not safe to eat as flies may transmit pathogenic bacteria. Thirdly, this study gives an insight to create awareness on food contamination by flies and to not take the matter lightly. Lastly, this study underlines the importance of food safety and hygiene. The problem regarding with the flies arises when Food and Drug Administration established a concern on the presence of flies around the food and food facilities. A research stated that flies are known as mechanical vector in spreading pathogenic bacteria. Moving forward, there are two objectives in this study. Firstly is to isolate and identify foodborne pathogens present from the interior and exterior body of flies. Secondly, to identify the effectiveness of common antibiotics to the pathogens isolated from flies. Listed in the slide shown are the literature reviews. Firstly, the filthy environment lived by the flies is also a suitable environment for pathogenic microbes to thrive. Secondly, living microorganisms need to be consumed by fly larvae to complete development as these microbes provide vitamin B or other nutritional factors. Lastly, Monyama et al. Stated that the gold standard of microbiological methods was by conventionally culturing and plating. The methodology was started with fly collection by using electric flycatcher. The flies were killed at -20 degrees Celsius for 10 minutes. The isolation of pathogens were started first from the outer body of flies. Sterile saline solution was used for washing of the outer body of flies. Then the washing solution was used to culture onto McConkey agar and Newton agar. Then the isolation from the internal bodies of flies was proceeded. The flies were crushed individually in sterile phosphate buffered saline. Next, the crushed fly in sterile PBS was centrifuged. The pellet was used for the inoculation on McConkey agar and Newton agar. After single colonies were obtained, gram staining and biochemical tests were performed. Then the antimicrobial activity test by using the Kirby-Bauer test was performed and the data obtained was analyzed statistically. Next, for the result and discussion, the first result that will be discussed is based on the table of the capture of blowflies. The disproportion of the number of flies between female and male was due to female flies had high tendency to land on microbe-rich substrates like meat, fish, and chicken for oviposition purpose. This has led to more female flies attracted to the bait and outnumbered the male flies. As for the second result is about number of blowflies that carried bacteria. As shown in the table, the number of blowflies carried bacteria externally was higher than the number of blowflies carried bacteria internally. According to a research, ingested bacteria subjected to successive bacteriolytic process. Meanwhile, the contamination of flies by bacteria were due to poor sanitary condition practiced by the hawkers like dirty floor and table and irregularly washing utensils like knife and chopping board. As for the third result, it is about the identification of bacteria isolated from the external and internal parts of blowflies. According to the table, the bacteria isolated from the external surface of flies were Staphylococcus, Bacillus, Neisseria, Proteus, and Citrobacter. In the internal part of the flies, the isolated bacteria were E. coli, Salmonella, Enterobacter, and Streptococcus. These bacteria are foodborne pathogens that become a great concern in causing foodborne illness, except for Neisseria and Bacillus. The presence of Neisseria and Bacillus along with Staphylococcus and Streptococcus were in relation with flies that shared the same environment with humans. As for the last result that will be discussed is about the antimicrobial activity of the antibiotics. Among all the antibiotics tested, ceftazidime and meropenem gave the largest diameter of inhibition zones. Citrobacter, Salmonella and Proteus were not very sensitive to the chloramphenicol antibiotics as the bacteria had significantly small diameter of inhibition zones. Nonetheless, all bacteria tested were inhibited by the commercial antibiotics, and the findings were in compliance with a study by Tufa et al., in which the researchers agreed that in non-hospital environment, they were less likely to isolate bacteria with antimicrobial resistant capabilities. In conclusion, the bacteria isolated from the flies whether externally and internally which have the risk to cause foodborne illness were E. coli, Salmonella, Enterobacter, Streptococcus, Proteus, Staphylococcus, Bacillus, and Citrobacter. Moreover, the common antibiotics that were most effective against isolated bacteria from the flies were ceftazidime, chloramphenicol, and meropenem. As for the recommendation, in the future, the molecular level of detection can be implemented to identify more bacteria in the species and subspecies level, as culture-dependent method is laborious and only certain types of bacteria can grow on the agar plate. With that, thank you.